Hello, everybody. My name is Vanessa Thomas, and I'm a grad student at the University of Texas, Rio Grande Valley. I'm here to talk about my current graduate research, which is looking at the Rhizosphere microbiome of guinea grass, which is an invasive species. Its synonyms include Panicum maximum, Megathis maximum, Eurocloa maxima, and other common names include bambusa grass. But for the rest of this talk, I will be only calling it guinea grass. So if you don't know what guinea grass is an invasive grass originally from Africa that was brought to the United States as cattle fodder as early as the 1800s. It has since then moved from cattle ranges out into native habitats here in Texas and has reduced the amount of biodiversity in grasslands. Because of this is actually an issue not only in our country, but has been seen in other countries as well, including Brazil, India, and Australia, where it is also considered a fire hazard in the United States and as well in Australia. So this is actually a nitrogen-needy plant. So it's very interesting to see it growing here in the semi-arid regions of South Texas, where our soils are kind of known to not have that much nitrogen. Therefore, we kind of believe that there is some type of relationship going on between rhizobacteria or some type of plant growth promoting organism, allowing it to take in that nitrogen that it needs to promote its growth. Other organisms that we think are having a relationship with guinea grass include arbuscular mycorrhizae, which is a fungi that grows on the interior of the root, which has been cited to help guinea grass grow in climate change conditions, which is like an increase in temperature. Because of this, there are also other citations showing that arbuscular mycorrhizae help guinea grass grow in areas to store more nitrogen and carbon than other grasses. Ectomycorrhizae is a fungi that grows on the outside of the root. So ectomycorrhizae has been known to help grasses by covering its roots with the fungal bodies and helping it grow in places and increase tolerance in terms of salt and intolerance in drought and heavy metal. This has also been shown to increase the biomass of the grass as well. This has been not only seen in guinea grass, but has also been seen in Phalaris aquatica, which is an invasive grass from Australia that has moved into California. Unfortunately, it has done something called legacy impact. So this is when the microbial community in the soil, because of the invasive habitants of the soil for a long period of time, the microbial community feels more obliged to have a positive relationship with this invasive grass more than with the native plants. The native plants and shrubs had a negative growth and relationship with the microbial community, and since then, restoration services and plans have really kind of stopped. So this is something that we can maybe see here happening in Texas, but as of right now, that is a current interest in my research. So my objective is really to look at the soil chemistry and see if there's any differences found between native and invasive grasses. Besides that, I want to look into the microbial community found between native and invasive grasses. And lastly, compare those differences and see if there's any alterations because of guinea grass and its invasiveness and changing the soil chemistry for its own benefit and being a really good competitor to native areas. So for my research, I went ahead and completed this by looking at different areas in the Rio Grande Valley. So I looked at 5 different parks here, all of which had different levels of hydration in the soil precipitation and soil type. This also had different ranges of invasiveness of guinea grass. For example, in Falcon State Park had the newest amount and lowest amount of guinea grass, whereas Benson State Park had the highest amount of guinea grass invasion. This allowed me to see if there is going to be any changes that are different in terms of Benson and Falcon, especially since Falcon has the newest amount of guinea grass growth compared to the other 4 parks. So for my methodology, I respectively collected soil samples from the rhizosphere, the rhizoplane, and the endosphere, which are the different parts of the roots, and did DNA extraction and quantified them in terms and amplified them, and I amplified them for PCR to see if there's actually any differences. And we'll be currently moving forward with sequencing to identify those individuals and see if there's differences between microbial organisms that are symbiotic in the roots of native and invasive grasses. Another thing I looked was the soil chemistry. I went ahead and collected soil from native and invasive grasses and looked at moisture, carbon, and pH and salinity. And I also looked into nitrogen and carbon analysis in the roots and the leaves of the grass. In terms of soil chemical results, we looked at pH, organic matter, which can be seen as leaf litter, conductivity, which can be seen as salinity, and moisture percentage that we saw in soils. So for pH, we can see that there was a flipped broad range between native and invasives. And the invasive, we can see that it can survive with a little bit of a lower pH compared to the native, which prefers to live in a higher pH. Also, with organic matter, we can see that there is a broad range of organic percentage found in invasive compared to native. This could be due to leaf litter. In terms of salinity tolerance, we can see that native grasses do not really do well in high salinity compared to invasives, which can withstand a pretty high amount of salinity, which can go back to the idea of there being a symbiotic relationship with the ectomycorrhizae. And in terms of moisture percentage, we can see that even though there are very same locations, the invasive grass had a higher moisture percentage compared to natives. In terms of further results, we can see that this data was collected based on location and on whether or not the subject was a native or invasive grass. So in terms of the invasive grass and soil carbon percentage, we can see that there is kind of like a mixed, where there are times where the invasive has a broader range of soil carbon versus the native. But in soil nitrogen, there is a very apparent significant difference between the native and invasive in terms of soil nitrogen percentage, showing that invasive does have a higher amount. There is leaf carbon, which was not very significant, whereas leaf nitrogen, there was a higher percentage found of nitrogen inside of the invasive guinea grass compared to the natives. So looking at this data, we can see that there is a cyclic type of relationship between the invasive grass and nitrogen. As we saw in the previous graph, that there was a high amount of organic matter. So in other words, we can see that this bunch grass is collecting nitrogen and therefore collecting it through the soil into its leaves and then putting it back into the soil and then putting it back into itself, allowing itself to continue using its nitrogen. But we understand that there might be, this is laying down the foundation of there being a organism that is helping it take in that nitrogen as well. So the following here are the images that we have here from our lab as preliminary microbial results. As we have already talked about, our vascular mycorrhizae, which is a fungi, has relationship with the guinea grass. Now, and from our lab, we have here pictures of the dark septate endophyte. So dark septate endophyte that promotes growth in grasses has already been cited through literature to promote in grasses in semi-arid regions. That literature support, followed by our pictures here, kind of shows that we are being put into the right direction in our research, that yes, that there is a relationship, and we hope with our further results in terms of sequencing that we can actually identify who these individuals are, and with our previous results confirm that there is that relationship. Now, so far in our project, we have already collected the DNA from the 3 different parts of the roots in both native and invasive grasses across the 5 different parks here in the Rio Grande Valley. I have already amplified them through PCR using 16S primers and have shown here that there are 550 base pairs that were amplified, and of course the bottom is the primer, kind of showing that we have successfully amplified some individuals, but through sequencing and through further methods, we want to go ahead and identify those individuals. Now, just to kind of like wrap up everything that we talked about today, so far in the research and our data that has been supported, that there is a difference between native and invasive grasses in terms of soil alterations. We have seen here that the invasive had already increased soil nitrogen ratio, both in the soil and in the leaves. There's a higher salinity tolerance, whereas natives, the only thing that they have is just that pH range difference. This is all pointing again to that direction that there is most likely some type of relationship, whether that be with rhizobacteria, mycorrhizae, there is something going on. And we hope to go ahead and finally identify these individuals and have that information available. But yes, thank you so much for everybody, patience and listening. And I would like to go ahead and thank my committee members, Dr. Miranda Torres Avila and Dr. Erin Schinzel, and of course, a big thank you to the Texas Parks and Wildlife for allowing us to go ahead and collect our samples at their sites. So yes, thank you so much.